A warm welcome once again to all ISPIC participants and teams to the 8th webinar of the Smart Protein Innovation Challenge India 2021 Inspiration Webinar Series. And this webinar is focused on the untapped potential of microalgae and seaweed in alternative proteins. I am Akshay Bhatt, the Cultivated Meat Science and Technology Specialist with GFI India, and will be hosting this exciting session today. We also have Nicole Roque, Innovation Specialist from GFI India, as a co-host for support. So before we delve deep into the webinar, some basic rules of engagement as usual. You as a participant can use the Q&A button to ask specific questions to the speaker, but also feel free to use the chat if you have anything else to share with the rest of the participants as well. Please do not use the chat for personal conversations. And post the webinar, we'll have a Q&A session for 20 or 30 minutes where participants will be given a chance to ask questions live. So if you could please press the raise hand button after the webinar, we'll allow or unmute you and allow you to ask a question directly to the speaker. So without much further ado, today we have with us Dr. Iris Haberkorn, postdoctoral scientist working with Professor Alexander Mathis, who heads the Sustainable Food Processing Laboratory, Department for Health Sciences and Technology, DHEST, ETH Zurich, Switzerland. Dr. Iris has a PhD in food science and technology from ETH Zurich, and during which she worked on microalgae bioprocess optimization to address terrestrial and space demands by applying nanosecond pulse electric field processing and microbial ecosystem management. Iris combines expertise in food processing and biotechnology, aiming to drive sustainable innovation in agri-food sector with a focus on single cell-based value chains. So a warm welcome, Iris. Sounds like space science to me.
Yeah, somehow a bit. Yeah, we also did space science.
Next up is obviously the innovation challenge would not have been possible without the generous support of our sponsors, our title sponsor Capri Global, our key sponsors NICE and Alcon Foundation, organizing partner CII Eco, supporting partners BIRAC, NIFTIM, CCAMP, AIC, CCMB, IIT Madras Bioincubator, Sign, and Able. Our ecosystem partners Blue Horizon, DSG, Fixie, Brink, Ankur Capital, Axelor Labs, ProVeg, Omnivore, Agni, IMSA Ventures, Big Idea Ventures, and Huddle. So the focus today is obviously microalgae and seaweed, and I think this basically plugs into track one of the specific RFP4 of track one, which is optimization tools for composition of growth media and for microalgae and seaweed, which is a specific bottleneck or a white space area in the space of smart proteins linked to microalgae and seaweed. So I'll not go into much of these details because we have this webinar and an upcoming webinar which will talk much more detail about feedstock optimization. So without wasting further time, I request Iris to please kickstart her webinar. So I'll stop sharing my slides now.
So then I can start sharing.
Yes, please do.
And going into presentation mode. Yeah, so I just jump right in. I'm very sorry for the delay, but I have had some technical issues, so to say. But I'm very happy to be here today, and it's a real pleasure and also honor being asked by the GFI to present our research here that we are conducting at the ETH Laboratory of Sustainable Food Processing under the supervision of Alexander Mathis, like you also mentioned before, and talking about the untapped potential of microalgae in alternative proteins. We are mainly working on microalgae.
Iris?
Yeah.
Sorry, you're still not on presentation mode.
Ah.
We can see your speaker notes.
Ah. I'm sorry.
No
How
worries.
do I? Wait, then I click stop share. How do I switch that?
Yeah, if you can just go to a presentation mode on your computer first and then share the specific screen.
Sure, good. I'm really sorry.
No worries.
But just a second. Share screen, so this one, perfect. So now you see the presentation?
Yes, perfect.
Perfect. Thank you so much. And again, sorry for any kind of delay and confusion. So still good. Then I want to jump right into the topic after spending some time on the technical issues. So as I said, I will be more going into the potential of microalgae, because in our group, we are mainly working with microalgae, less with macroalgae or seaweed, how it's commonly called, and here working on potential technologies and applications on how we can make those microalgae grow more efficient. But just to start into the topic, I wanted to highlight some recent relevant publications on the food system sustainability, just to highlight the urgency and also the relevance of the topic. And all these articles, they're just from the last year, from this year, or highlighting that there's a clear need for a multidisciplinary collaboration. These publications are all open access. And like I said, there's a clear need for multidisciplinary collaboration. So we food scientists need to engage on a multi-stakeholder level here too. And we also need to focus on social innovations and behavioral change, which have more impact than only technical innovations. However, technical innovations can support social innovations and a behavioral change. And here in Switzerland, we have the Swiss National Food System Summit Dialogue, which is called From Challenges to Actions, working with delegates of the Federal Council for the 2030 agenda, and also the Swiss National Science Foundation, or working together with FAO or WIS Academy to work towards the Sustainable Development Goals and being able to interact on a multi-stakeholder level here. And to just provide an overview on our group and what our group is actually doing, I prepared the following slide, which shows you that our group is actually part of the global bioeconomy. And we're working on different topics, and our three main objectives are food security, side stream utilization, and healthier and safe foods. And we approach those objectives by applying thermal, electromagnetic, and mechanical processing technologies that we align with an approach that we call life cycle sustainability assessment to see whether these technologies and novel raw materials that we are investigating really more sustainable than currently employed solutions. And we base our research on what we call advanced approaches, which means that we are using innovative raw materials such as microalgae, but we are also working with insects and working in a complete biorefinery approach. And here, our group covers the whole value chain. As I mentioned, we are going from those novel raw materials, which we consider microalgae or insects to be, and then we cover the whole value chain, starting from the primary production of those novel raw materials, over processing them. So we are looking at possible ways on how can we make them grow more efficient and also more sustainable. How can we disintegrate or separate these novel raw materials in order to get certain components out of interest for us, for instance, food applications, but then also characterize their techno-functional properties. For instance, if we think about proteins, it is really important to understand those structure process property relationships if we really want to apply those proteins, for instance, in emulsions or foams or other food products like meat analogues. And then we also go via the processing. So we use those novel raw materials, complete microalgae or extracted components thereof, for instance, the proteins. And we want to see how do we need to modify processes to obtain desired products, of which we then also assess the quality, which is also, of course, really important because we need microbial and chemical safety to then, in the end, go to the consumer and see, okay, how are those foods taken up by the consumers, combining it also with medical and consumer science here, because if we talk about microalgae, a high value component ingredient, we need to make sure that these components that we find in the microalgae are also bioavailable or bioaccessible. Therefore, also combining our research, starting from the primary production, which influences a lot the composition of those novel raw materials, to doing consumer and medical studies. And the challenge that we are actually all facing, you might have already heard it, is that we are facing a growing world population that is estimated to increase to around 9.7 billion people by 2050. And more people will be living in cities, which poses challenges associated with food security. However, our current agricultural and food systems are challenged to sustain a growing world population, and food systems are among the largest drivers of global environmental change. They foster climate change and cause the Earth systems to transgress beyond their planetary boundaries. Resource-intense agricultural systems have caused water scarcity, soil depletions, and high levels of greenhouse gas emissions. And a recent Nature publication showed that our food systems account for around 1/3 of global greenhouse gas emissions. And food systems have contributed to biodiversity loss and interfere with global phosphorus and nitrogen cycles. In turn, climate change causes extreme weather conditions, which lowers agricultural productivity and the nutrition quality of foods, which eventuates then in rising prices for food and tightens its access for people, which is especially of concern if we think about low-income groups. In that context, it appears controversial that globally around 1/3 of the food that we produce is wasted or lost with blind spots along the value chain, while it is estimated that approximately 2 billion people globally already show now nutritional deficiencies and 800 million people suffer from hunger. Hence, these trends pose challenges that affect our traditional understanding within myriad domains, and that we need to foster a shift toward more sustainable and climate-smart food systems. And here the UN have set up the 17 Sustainable Development Goals to foster such a shift to more climate-smart food systems. And here we can say that food systems are at the heart of at least 12 of the 17 Sustainable Development Goals. However, if we think about the UN Sustainable Development Goals, a concrete toolkit is missing on how to implement or foster changes. These goals are just posing, or they're just guidelines on where and how or that a shift should be fostered. And here, as I mentioned before, the life cycle sustainability assessment is a quite good approach. A holistic approach that integrates previously siloed food sustainability assessments. And here we could show with a first global analysis that quantified the status of national food system performance of 156 countries, employing 25 sustainability indicators across 7 domains, which are nutrition, environment, food affordability and availability, social, cultural well-being, resilience, food safety and waste. And we could show that different countries have widely varying patterns of performance with unique priorities for improvement. And we could show that high-income nations score well on most indicators, but poorly on environment or food waste and health-sensitive nutrient intake indicators. The study also showed that we need to foster a transition from animal foods towards plant-based foods, which would improve the indicator scores for most countries. And these matrices or the life cycle sustainability assessment can help nation-specific, or these nation-specific quantitative results can help policymakers, for instance, to set improvement targets on specific areas and adopt new practices while keeping the track of the other aspects of sustainability. So really supporting a guideline on how to perpetuate these sustainable development goals and which targets we should focus on as kind of guideline. And with this multi-indicator sustainability assessment, we also did a case study on pulse integration into food commodities and how it could affect the sustainability indicators. And here we used yellow pea as an example and incorporated it, for instance, we simulated how it would affect sustainability profiles. If we, for instance, add 15% for pan bread, substituting wheat flour by those yellow peas, 53% for the breakfast cereal, and 30% for pasta. And we could show that incorporating, for instance, those low-cost pulses, such as yellow peas, that are quite nutrient-rich and low in fertilizer requirement into daily food items, can improve the nutritional and sustainability profile of national diets for the different scenarios, bread, cereals, and pasta. And the results also showed that partial replacement of refined wheat flour with yellow pea flour increased the nutrient balance score of pan bread, breakfast cereals, and pasta by the shown percentages on the left here. So 11%, an increase of 70%, and an increase of 18%. And it decreased the life cycle carbon footprint in all scenarios, and most for pasta. In this study, the nutritional and greenhouse gas data were combined as the nutrition carbon footprint score, so the NCFS, which is a novel indicator that reflects product level nutritional quality per unit environmental impact. And the results showed that yellow pea flour increased the NCFS by 15% for pan bread, by 90% for cereals, and by 35% for pasta. Hence, we see that integrating pulses into food commodities can improve several sustainability indicators, such as the nutrient balance score, the life cycle carbon footprint, and nutrition carbon footprint score. And if we think about alternative proteins, the topic of today are microalgae. However, pulses are considered as kind of benchmark for plant proteins if we think about short-term and large-scale application. If we now want to go for other single-cell or other protein sources, single-cell-based protein resources, such as, for instance, also cultured meat or microalgae, we are talking here about mid to long-term solutions, and microalgae are considered as alternatives to benchmark proteins, to plant proteins, while lab meat, for instance, is considered as an alternative to benchmark animal proteins. And why are microalgae so interesting? As already mentioned before, we have an increasing world population, and food production will need to increase to 70% or by 70%, with more and more people living in urban areas, which means that there's no more significant free arable land, as we are also facing deforestation. Therefore, we need alternative resources grown on non-arable land, which could, for instance, be single-cell-based technologies such as microalgae. And microalgae can tackle three core sustainability principles, namely efficiency, sufficiency, and substitution. Microalgae turned especially interesting as they have a couple of advantages. For instance, if we grow them photosynthetic, so which means that we are harnessing light for creating biomass, biophotosynthesis, we can use the microalgae for CO2 uptake. Microalgae also have a high protein content, which is up to 70%, depending on the species that we use, whereas also not only the protein quantity is really desirable in microalgae, but also the protein quality, and I will show a figure on that later. Microalgae, as said before, can be grown on non-arable land. They have a high biomass productivity, which is, for instance, 5 to 10 times higher than that of conventional food crops. And we can, with microalgae, we can have a variety of different applications. We can, for instance, extract components, or we could use the whole microalgae. And of course, microalgae are also very nutrient rich. So we could use lipids, proteins, pigments, vitamins. So they're really a powerhouse here. And as just mentioned, microalgae are really attractive as they don't require arable land for their cultivation. And to illustrate this, I have the following figure here. So what you can see here now is a comparison of different protein sources. So we have conventional protein sources such as beef or dairy cow, where you see the land requirements are really high for getting a kilo of proteins. But also if we compare microalgae to other plant-based proteins, for instance, soy or pea protein, we range lower than those other plant-based protein sources, with 1.6 square meters per kilogram of protein. And as microalgae have a very low land requirement for the production of proteins, and their cultivation is independent of arable land, we could, for instance, also consider cultivating them in an urban surrounding or an urban environment. And I have two examples here. So there's one house that is called the Algae House, which was installed in Hamburg. So this house actually really exists. And here the idea is to cultivate microalgae in the facade of this house, which is completely surrounded by those bioreactors, harnessing CO2 for biomass formation or for microalgae cultivation. And another example is the startup Analogues, which you can see on the bottom left picture here. They're cultivating microalgae in buckets on rooftops in Bangkok. And if we then couple those urban food production systems with an urban food processing technology, as for instance, you can see on the right here, which is a downscaled system, we can then go for urban food production coupled with food processing using the microalgae, not requiring arable land anymore. But of course, if we go for industrial scale production of microalgae biomass, it currently looks different. You might have already seen it. So we can cultivate microalgae in different modes. For instance, these open ponds that you can see in the middle here, or also in a more controlled manner in photobioreactors, for instance, if we want to go for pharmaceutical applications. Both are using photosynthesis of the microalgae, so using light for and CO2 for producing biomass. But if we cultivate microalgae, we don't only have this photosynthetic cultivation mode, we also have what we call heterotrophic cultivation of microalgae, so under the exclusion of light, using organic substances such as glucose, but also sometimes fructose or lactose are used for biomass or for generating biomass. And what you can see here, I have it in euro prices, but it's a price comparison on how much it costs to produce a kilogram of microalgae on the market. And what we can see here is that if you go for the heterotrophic cultivation, because we don't need all this energy for light supply, we are at 1.7 euros per kilogram. And then if we go to a similar, very controlled system, so the photobioreactors or the tubular systems, you can see that with increasing process control and also increase in energy needed, for instance, for supplying light, the price per kilogram of microalgae increases significantly to 19.6 euros per kilogram. This also illustrates us that the prices for microalgae and for microalgae biomass are currently still very high. And if we want to be competitive, we need to decrease the prices for the microalgae biomass, especially if we think about creating some kind of meat analogues that should be a food commodity for consumers. So here we still need to work on process optimization, both in the upstream part and in the downstream part. Especially if we consider that microalgae are very nutrient dense or nutrient rich. And here you can see the amino acid profile comparison between microalgae and conventional sources of protein with those recommendations that are provided by WHO or FAO. And you can see that the amino acid composition, so not only the quantity of proteins in microalgae, but also the quality is quite good. And it compares quite well if we consider or compare it with other conventional traditional protein sources such as egg or soybean. And they also go quite well with WHO and FAO recommendations. However, what we also see, or what a study that was conducted in our lab could also demonstrate, is that the protein or the macronutrient composition of microalgae can be very diverse. It depends on the species and the cultivation conditions. The study also showed us that the protein content can be up to 70% with this well-balanced amino acid profiles that are in line with the WHO and FAO recommendations. And more nutritional benefits. So while microalgae are also beneficial, for instance, if we compare them to yeast or other single cells such as bacteria or what we see is that they have more nutritional benefits than many other single cell approaches. For instance, a beneficial ratio of omega-6 to omega-3 polyunsaturated fatty acids, but also a low nucleic acid content, yet high contents of beta-carotene, polysaccharides, and vitamins, such as, for instance, vitamin B12, which is very interesting if we consider plant-based meat alternatives for vegans, for instance. However, if we want to make microalgae a very attractive solution for the future, we will need an alignment of the upstream and downstream processing in the biorefinery concept of microalgae, which means that in the upstream processing, we need to improve efficiency of microalgae biomass production, increasing the yields here in a sustainable manner, and that in the downstream, we harness all the valuable components that are within the microalgae with novel ways of harvesting and also performing such kind of what we call a cascade extraction of the valuable components, which means that we extract proteins, but also lipids, antioxidants, vitamins, so all the valuable components. And only if we make use of all those components or the whole biomass as such, microalgae can really be a sustainable solution. And using it as a biogas or fertilizer, how it's also often done, should only be a very last step in the usage of microalgae. And to give you some insight into the research that we are really conducting and at ETH Zurich here and the Sustainable Food Processing Group and how our labs look a bit and how we tackle this challenge, I provided a small video. I hope it works.
Hey, hi, Iris. I think the video is working, but the audio is not. It's not audible basically, but I think it's still fine.
Pardon?
The audio, it's not audible. The video,
okay.
The audio of the video is not audible. basically.
Ah, okay, well, then probably I just leave it to you to watch it if you're interested. I mean, I can keep on going because the text is there, right?
Yeah,
so
please
you
go
can,
on. Yeah.
Yeah, so you can just follow it. Great. So I hope, although the sound didn't really work with the subtitles, you could get some kind of impression on the topics that we're working on with the microalgae and also what we're doing in our lab. So working on the upstream and downstream processing of microalgae. And I will now go ahead to go more into detail what we are really actually doing in our labs and present you some results that we figured out over the course of our research. And what we could see here, if we look at the upstream processing first, was that if we apply innovative microalgae upstream cultivation, so cultivating, for instance, Chlorella vulgaris or Oxenochlorella protothecoides, as in this example, in different cultivation modes, so photoautotrophic, mixotrophic, or heterotrophic, we can play around with kind of the pigment content, so decrease chlorophyll or increase carotenoids, and then also achieve different colors in final applications, for instance, in foods, cosmetics, and bio-based applications, which is, for instance, especially interesting if we think about the consumer acceptance of final products, because the studies that have been conducted so far show clearly that consumers would be less prone to eating what you see in the top here, so the phototrophic or the emulsions based on the phototrophically cultivated microalgae. Consumers are less prone to consume those products because they're very green, and also if we process them, for instance, in meat substitutes, they might turn gray or not very appealingly brownish. So for us, it's really important to also work on this upstream part, the upstream cultivation of microalgae, to play around with those components like pigments to define and foster some downstream application in a final product. And if we look at different cultivation modes, we can clearly see that we can get or foster different colors applications for products that are appealing for consumers. And if we think about upstream cultivation of microalgae, what our group is especially focusing on, and what is also mentioned before, is post-electric field processing. And here we have different people working on those on this topic to foster microalgae value chain innovation, both in the upstream part and in the downstream part. And I will explain you on how we aim to, or how we are applying this very innovative, novel technology, both in the upstream, but also in the downstream cultivation of microalgae, to make the biorefinery or the value chains more climate smart and more eco-efficient, and to finally decrease costs of microalgae production. So here in the upstream part, we are mainly focusing or using the PEF processing, or PEF, how we call it, for microalgae growth stimulation. In the downstream part, we are working on the extraction of molecules using post-electric field processing, which is a very nice approach if we, for instance, compare it to other commonly used or industrially used downstream methods for harvesting or component extraction, as for instance, high pressure homogenization, which completely destroys the whole cell, but also intracellular components. So then destroying or losing the techno-functional properties of those components. And if you use post-electric field processing, as I will show later, we can extract the components while maintaining their techno-functional properties. And to give you an idea or a better understanding on this post-electric field processing technology, you can see the basic principles of this PEF action displayed on the slide here. So actually, if we apply an electric field to a cell, it means that we have an accumulation of charges at plasma membrane level. And this accumulation of charges induces pore formation in the plasma membrane because, for instance, it induces reactive oxygen species or the migration of water molecules through the membrane, but also a flip in the lipid molecules that we can find in the plasma membrane to rearrange and form pores. And depending on the processing window applied, we can trigger those pores that they're either reversible or irreversible. So reversible means that they open for a millisecond or sometimes for a nanosecond, which opens the window for extracting components, for instance. And irreversible means that we apply a very intense processing window and completely destroy the cell or disintegrate the cell. However, if we want to go for the upstream processing, what is quite important here, and you can see that the principle or the basic understanding that you need for understanding on the bottom right graph here. So this part in the green and in the red thread. And what you can see here, so the dashed line is the transmembrane potential of the plasma membrane surrounding the cell. And the straight line is the transmembrane potential that can also be applied to organelles, so on intracellular level. And what we can see here in the right dashed graph or part is that actually the transmembrane potential on the plasma membrane is higher than on intracellular level, which means that we are creating those pores mainly on plasma membrane level. And this we do by a technology that we call Microsecond post-electric field processing. So we are applying pulses in the microsecond range. However, you can apply pulses that are way, way, way shorter, so in the nanosecond range. And then you get into this red thread here, where you see that the transmembrane potential of the organelle exceeds the one of the plasma membrane, which means that we are primarily triggering intracellular structures of the organism, of a single-celled organism, and having less effects on plasma membrane level. And why is this important? It is important because we can exactly trigger this processing window with the devices that we have here at ETH Zurich. And what we can trigger with it is an intracellular abiotic sublethal stress response in single-celled organisms. And here we could achieve a growth promotion of different kinds of microalgae by inducing those pulses, but also of bacteria and the yeast Saccharomyces cerevisiae. And you can see that by applying these pulses in the nanosecond range, we induce intracellular sublethal stress and trigger the growth of Arthrospira platensis, which most people know as spirulina, significantly, but also of Chlorella vulgaris, both cultivated photo and heterotrophically. And these are one of the two most industrially relevant microalgae, but also of the yeast. So we can see that this nanosecond postelectric field processing is a quite interesting technology, and it goes over different organism domains. And what is also interesting for us is that we can just treat the cells in batch mode. We don't need to supply any more nutrients or any more treatments. The energy demand for this technology for applying the pulse is really low, but still we can increase the biomass that we obtain up to 17, 18%. And what was also very interesting for us to see was that if we apply those pulses in spirulina, we can significantly increase the phycocyanin content in Arthrospira platensis. And why is that interesting? Phycocyanin is a high-value component. I will show you more applications for phycocyanin later, where we can achieve quite high prices, and it's the only naturally occurring blue pigment for the food sector globally. So it's quite interesting for us, without any additional needs for nutrients, to significantly increase the content of this pigment, of this high-value component in Arthrospira platensis. And to now go from the upstream to the downstream processing, I will show you some examples for the extraction of molecules. And as I said before, if we now think about the extraction of molecules, we are not on intracellular level anymore, but on plasma membrane level. And here we are focusing on creating non-permeable pores or irreversible pores, how we call it. And the beauty of using Postelectric field processing in the microsecond range here is that we can extract the components while maintaining their techno-functional property. And here what we came up with is something that we call microalgae milking. So which means that you do some kind of cyclic protein. Also, we were focusing on the proteins because we want to focus on meat analog creation. And what we could show was that we can do some cyclic protein extraction from the microalgae, so inducing reversible pores on plasma membrane level, getting out the components of Chlorella vulgaris in this case, with the specific extraction cycles, but then the algae maintained their viability. So you can just take them and then cultivate them further until you get to the next extraction cycle. On what we basically did was that we have the microalgae that we prepared, then which you can see in the left here. Then we did the PEF treatment in the microsecond range. We incubated for 24 hours. We separated the protein, so the supernatant, from the remaining biomass. And then we looked at the extracted proteins. How much is it? How often can we extract? But also at the revitalization ability of Chlorella vulgaris. And if we talk about the proteins, of course, we also need to consider that if we do the cyclic protein extraction, we are targeting a specific group of proteins, namely the free proteins or the soluble proteins. And if we go for PEF processing, as just said, we mainly extract those free proteins from the cell, but not internal or structural proteins. And if we compare it, for instance, to high pressure processing, which is commonly applied for extracting components from microalgae or for destroying their structure, Here we can see that with HPH or high pressure processing, we can extract all the proteins that we can find in the microalgae. And here we were combining, or we were trying different processing parameters, and you can see that if we apply different electric field strengths, so different intensities of the PEF treatment, we can also extract different amounts of protein here, which you can see on the top left graph displayed. So if we apply a very harsh treatment, of course, we can extract most protein from the cells, but what you can also see on the bottom left graph is with the red line, if we apply a very high electric field, so a very intense treatment, the revitalization ability of the microalgae is not very good. This means that we can extract a lot of components or proteins here, but it's also very likely that we just destroy the microalgae with this very high electric field. Therefore, if we want to extract proteins from the microalgae, but keeping their viability for having a continuous or repeated cyclic extraction, we should go for lower electric fields, like for instance, 15 kilowatt per centimeter, where we also have a high recovery rate of the microalgae. But of course, we also see that the protein extraction is quite low. So if you look at the graph, it's like around 0.05, 0.1 gram per liter protein. And of course, this was just the first study to see whether this works, whether we can revitalize the microalgae. And we need to improve this further, this technology, to really being able to use it as a downstream processing approach for the microalgae. And if we compare it with high pressure processing, we can also clearly see that the protein concentration that we get out of the microalgae is way, with the Postelectric field processing, is way lower. So PEF treatment is not competitive to high pressure processing or high pressure homogenization yet. However, the Postelectric field treatment has limited effect on the cell's morphology. So we further need to improve this process here in order to obtain higher values or higher percentages of protein that we can isolate from the microalgae, because we can maintain the structural and techno-functional properties of the proteins that we extract. And Postelectric field processing is also very interesting because it's a low energy demanding technology. And if we think now about those proteins going more into the downstream part, what we also aim for is we have a certain functionality. This is why we are using those proteins. And we also want to create value from those proteins with final applications. And here we go for a biorefinery approach. And such a biorefinery concept can be set up with the goal to maximize its protein production. And this is only valuable in my point of view, or in our group's point of view, if you can reason this with an identified functionality. And for this, I want to highlight what you can do with microalgae in extrusion, emulsion, and color stability, looking again at the phycocyanin that I've been talking about before. And if we go for the high moisture extrusion, we in our group were trying to create a meat analog, a hybrid meat analog based on using microalgae, because we can have an added nutritional value if we include microalgae into plant-based meat analogs. And here we used extruded soy concentrate in heterotrophically cultivated Auxinochlorella prototicoides, which has a light yellow coloration, and extruded it into fibrillary textured meat analogues. And by using light yellow microalgae, a consumer adverse visual appearance can be prevented. So as mentioned before, in the upstream cultivation, where we focused on different modes on how we can cultivate the microalgae, here we use really this golden chlorella, how it's called, or yellow chlorella, because we don't have this greenish appearance from the chlorophyll. which is not perceived as not very accepted by the consumer. So therefore we use this yellow kind of microalgae to increase consumer acceptance in final food applications. And what we could see was that by integrating microalgae in these meat analogues, we observed a significantly improved nutritional profile of the extradate by incorporating, for instance, vitamins such as vitamin B1, vitamin B2, B3, B6, and E. And here we could also show that over 95% were retained in the final product after adjustment during high moisture extrusion. And this study was really interesting for us as we were the first ones globally to produce this microalgae-based meat substitute. But what we could also see was that we used up to 50% spray-dried microalgae biomass, which affected the formation of fibers, which we could balance by reducing, for instance, the moisture level. In some analysis of those meat analogues showed, which we did with optical impressions, that we could quantify with specific mechanical energy input, so SME, which you can see on the left graph here, and texture analysis on the top left graph here. What we could show was that with increasing microalgae content, we decrease friction and equally reduce texture analysis. And we assume in our group that this process is fostered by the microalgae that we include into the meat analog in two different ways. So the first way is that there's a higher fat content. If we compare it to pure soy protein concentrate, if you look at the table on the top right here, we can see that microalgae have a way higher lipid content than pure soy concentrate. And additionally, we could show that intact microalgae cells in the extradate were existing, which you can see on the bottom right graph here. So a microscopy analysis of the extradate. So we could show that there were still intact microalgae cells in the extradate, and that these intact cells could function as kind of inert filling material, which might hinder structure formation. In addition, microalgae protein is captured intracellular because we were using whole microalgae cells without any treatment. So the protein that is usually contributing to such extrusion process or products made from extrusion usually contribute with texture formation. However, if you keep the proteins within the microalgae, of course, they can't contribute to structure formation. So concluding from the study is that we could show that microalgae can be used for making meat analogues with a higher vitamin content. However, we could also see that there's some room for improvement. For instance, further studies looking at disintegrating microalgae before using them in an extradate to see whether we can get a less fibrous structure that is more similar to meat. And if we go now from the extrusion to a more microscopic level, so to the emulsions, which are, for instance, important in the food sector. If we look at, for us, always an example is mayonnaise, but also if you go to cosmetics industry, if you want to create vegan products, so vegan creams or also foams, microalgae can be very interesting as they provide proteins with interfacial properties, surface stabilizing properties that could function in vegan applications. And in our emulsion research, we followed two questions. The first question was, can we use microalgae proteins as emulsifiers? And the second question, if we can use them, is it worthwhile to purify the proteins, or could the entire microalgae be used? Because usually the purification process is quite intense. So it's a very valid question to ask, do we really need to apply this very extensive purification process to make emulsions work, or can we just use the whole microalgae as such? And what did we do? We applied the Atrospira platensis, so the spirulina, and we created different extracts by homogenizing these spirulina cells, and we centrifuged them to get a soluble extract. And in the end, we precipitated the proteins at the overall isoelectric point and diafiltrated the obtained protein isolate. And we then looked at these extracts, so the whole microalgae cells and the different extracts that you can see displayed in the graph on the right here, with two questions. The first question, can we stabilize oil-water interfaces with them, which would allow us to use them as emulsifiers in emulsions replacing animal proteins? And what changes when we purify the crude extracts that contain all components of the algae into protein isolates? And to test how the extracts diffuse to the oil-water interface and form a physical network, we used an interfacial shear rheology. And the rheometer is therefore equipped with a biconical disc that is positioned exactly at the interface of oil and water. And from small oscillations, the physical response of the interface can be then recorded over time, which you can see as an example or a graphical illustration on the top left here. And this leads to the result shown on the right, interfacial modeling over time. The protein isolate, what you can see was that the protein isolate directly adsorbed to the interface, and it forms a viscoelastic network. And the soluble extract, on the other hand, depicts a lag phase of several hours. And we could also see that final values, even after 20 hours, were still inferior to the protein isolates, which means that the purified protein establishes a faster, a stronger network at the oil-water interface, which is an important step for us to replace other protein-based emulsifiers. Yeah, and to wrap up these findings, so on how we studied on purifying proteins from spirulina biomass, changes and effects to the potential of the biomass to stabilize emulsions, we could, to wrap up, see that we did a comparison between what you can see here on the left, the entire microalgae biomass, and on the right, the isolated protein from spirulina or Atrospira platensis. And what we can see was that, I mean, by purifying 4 protein from the whole microalgae crude extract or from the whole microalgae, obviously there was protein yield lost. The emulsions could be stabilized successfully with both biomasses. However, smaller droplets could be stabilized with equal amounts of protein for the purified section, indicating that the layer thickness decreases for more purified proteins. And simultaneously, what we could show by interfacial measurements was that we could observe that the network at the oil-water interface strengthens with more purified protein. So overall, or to conclude from those studies, the protein functionality in terms of emulsifying efficiency increases by purifying the protein. However, what we also need to consider is that we purify this protein and we observe stabilizing abilities, but the increased functionality per protein that we can observe here has to be balanced against the much lower yield of protein that we obtain if we extract the components out of the microalgae. And to go into even more microscopic levels, so looking on the phycocyanin, so the blue pigment that we can see from, or that we can obtain from spirulina. It's, for instance, applied in Smarties or in ice creams, but it also finds applications in the biomedical research because of this nicely blue color properties and its fluorescence. And to give you a better insight in what phycocyanin actually is, it's used by the microalgae as a light harvesting complex by spirulina, but also by other cyanobacteria. And if we zoom into the spirulina cell at the thylakoid membrane, which is actually where the photosynthesis happens, which you can see displayed on the right here, we find these light harvesting complexes, or the phycocyanin, at two main components are involved. So the C-phycocyanin and the allophycocyanin, they absorb light at 620 and 650 nanometers respectively. So 620 for the C-phycocyanin and 650 for the allophycocyanin. And these are wavelengths which other pigments such as chlorophyll or carotenoids don't absorb given cyanobacteria biological advance and reasoning for this blue color impression. And the absorbance is kind of an interplay between the phycocyanin's protein subunits and the actual chromophore, the phycocyanobacteria. And the chromophore is, for instance, bound to the protein. It is forced into an extended state. And this and the close proximity to other chromophores results in the blue color impression that you get from the phycocyanin. And once this interaction is disturbed, as for instance by a protein denaturation, the chromophore switches back into the thermodynamically favored cyclic state and the color is actually lost. And exactly this phenomenon, if you want to apply it for food application where you probably need to heat, for instance, if we apply it in the Smarties or other forms, we might need to heat the phycocyanin. So here you can see again an example for the extended in the cyclic state. So this behavior under thermal conditions was exactly what we were interested in the phenomenon that we investigated in detail. And what we investigated was the short time thermal treatments, the color degradation, and therefore we used a microprocess engineering platform, which was donated to us generously by Nestlé, which is a continuous heating system with a high service to volume ratio, where we are able to control or to have a controlled increase and decrease of the temperature in a very, very quick mode and a very controlled manner. And what we could show was what you see on the graph displayed on the left here, that the color is lost within seconds. So yeah, there's a very steep decrease if you just look at the left-hand side of the graph at around 70 degrees, and that this degradation is best described as a biphasic process. And then we have this very steep decrease followed by a tailing. And in a follow-up study, we could then further purify the phycocyanin extract by a stepwise ammonium sulfate precipitation and subjected this phycocyanin to different scanning calorimetry tests and again to high temperature short time treatments. We connected it with absorbance, fluorescence, and circular dichroism spectra, and we could allocate individual denaturation temperatures to specific quaternary phycocyanin structures. And moreover, we could show that the chromophore and the secondary protein structure collapsed simultaneously. So structure and function of the phycocyanin go hand in hand. And what these findings allow us is to have a better understanding and better process control when employing this high value component in food and in other industries. And as two concluding slides, I would like to highlight to you our new project that we will be starting in our new lab at the Singapore ETH Center, where we try to combine all those findings that we get from the up and the downstream processing. So the cultivation of the microalgae, the Postelectric field processing, but also the characterization of techno-functional components from the microalgae to produce products, final products, applications that are accepted by the consumer, which will be the aim of the project here. And we will be building up a new lab in Singapore for doing that. And here the aim is the urban single cell protein production, working with microalgae and working on the up and downstream processing. And here we are applying a whole value chain approach as also the principle of our group. And we want to combine existing technologies and processes with low technology readiness level with emerging microalgae concepts. And we use several innovative aspects here, such as, for instance, 3D printing, extrusion, but also the postelectric field processing. For instance, we use heterotrophic microalgae cultivation on liquid food industry side streams to create a circular economy approach. And we want to establish a holistic postelectric field-based biorefinery focusing on growth stimulation and gentle disintegration for being able to foster the economic viability of microalgae by decreasing production costs and create a circular, more sustainable urban food production using microalgae. And for food production, we then want to focus on extrusion and 3D printing in iterative processes via reverse engineering principles. And we want to combine it with consumer research, which is a very important point for us, because if you don't know what consumers want, you can't really tailor a process. So our aim is really to include consumer insights and ask for consumers what they want, and also present them final products to understand what is triggering consumer behavior, consumer purchase behavior, why or what do they or why don't they like our project, or not our project, our final products. And with that, I'm at the end of the presentation or the webinar. I want to thank you so much for your attention and for listening to the research or to some of our research results. I hope it was inspiring or interesting. In that context, I would also like to acknowledge all our collaboration partners, sponsors of the research. And I'm open to answer any questions. Of course, you can also always write me or other people from the group. Just search our homepage, our webpage. We are a very diverse group working on insects, postelectric field processing, high pressure processing, on bacterial spores. If you have questions, just feel free to contact the people. And with that, I would like to thank you, and I'm open to questions or the Q&A section.
Yeah, thank you very much, Iris. Absolutely, it was insightful, very interesting, and also inspiring. We have a ton of questions, probably. I think a few were answered during the session, but we can go one by one. So Bhavesh Kumar, one of our participants, has a question for you, which reads as, which is the best microalgae culture suitable for alternative protein sourced for meat analogues, and how to overcome typical algal characteristics, smell, recovery, quantities, and sustainable production when compared to plant proteins?
I mean, so if you want to go for meat analogues, I think it's always, I mean, if you go for food production in general, there are only 7 kind of species that are allowed, right, for microalgae, and it always depends on what you want. So your final application is also triggering what you choose as microalgae. But what we just think is that, or what also studies from ETH showed, was that microalgae that are cultivated in heterotrophic mode, so without chlorophyll, without pigments, are better accepted by consumers. So I think this was the first part of the question, right? So which microalgae we would be choosing. And as I said, there are 7 options currently that you can use for food application, which is chlorella, spirulina. And if you look at bulk production, for us, the chlorella or oxeno-chlorella was the most interesting. And if we now go, so the second part of the question was about the meat analog, right? How it compares in texture and also in taste and smell with meat or other plant-based resources. So I can't really give you a 100% answer because we didn't do sensory tests on the microalgae meat analogues yet. Because we couldn't get food-grade biomass at that point. So this will also be something that we will be focusing on in the Singapore project to really get consumer studies in sensory science. But of course, there was also a very high percentage of soy in there. So the main smell perception that we could already get was mainly soy, because I think a main concern with microalgae is often it's tasting fishy, but we didn't smell anything fishy. And yeah, but I can't really give you answers on that because we didn't test it. We did a comparison on texture with meat, so poultry, and only soy. Yeah, but I think compared to meat, it was way more fibrous. So also what I mentioned in the presentation, that we need to now go on and check or see how disintegrating microalgae could work to get a better structure that is more meat-like here by releasing more proteins, because it was just very, very fibrous. I hope this answered the question.
Yeah, I hope so too, but thanks for answering the question. Obviously, I'm not an expert in this field, so that's the reason why I said that. but yeah, we can go to the next question now. Outdoor production is cost effective, but the surety of quality and probably the surety of the quality and quantity in changing climate conditions that cannot be managed. How do you think we can overcome that?
Yeah, I also see the point. So I think there's one. So a lot of people go for biomass production in such called open ponds, which I also showed. But the disadvantage here is that that you have a lot of contamination, or you can get a lot of contamination because you have just birds flying around and also bacteria. And I think there are downstream processes in place that take account for this increase in contamination. After the harvesting, I mean, there are also inactivation hurdles, inactivation steps involved here. What I also might see a concern is if you get some microalgae pathogens into the culture. I also already heard that a lot, that you can just get a microalgae virus into the culture and it just breaks down. So there, I didn't read it in literature yet, but or not to a very big extent. But there's some research is done on doing something like probiotic or protective cultures around the microalgae. So you could, for instance, apply a microbiome around the microalgae that is protecting them from getting pathogens in or other bacteria in that you don't want, and your bacterium or microbiome that you actually put with the microalgae could foster its growth or stabilize the culture while it's still very controlled. And there are also things done, or research is done in investigating selective inactivation technologies. So for instance, also using postelectric field processing. You can use for selectively inactivating bacteria while maintaining microalgae viability. So there's also research done here. But of course, if you want to go controlled or pharmaceutical application, we go for the bioreactors, which are still very expensive owing the process control. And here we would need to decrease the costs of microalgae production using more sustainable energy resources, for instance.
Yeah, thanks, Alice. Probably we have a few more questions. We've got like 8-10 minutes left on the clock. So let's go through it very quickly. What's the most suitable method? Probably you've answered this partially, but what's the most suitable method that can be used for extraction and purification of protein and lipid components present in algae among existing methodologies, basically, without much protein degradation also in terms of yield?
Yeah, so high pressure homogenization is done. Then just normal spray drying, ultrasound is done, plasma is done, I think, but it's not very well established. Or the post-electric field processing. It always depends on what you want. Whether just destroying them is okay. You can just go for drying, spray drying, high pressure homogenization. Or if you want to keep components, go for cold plasma or for post-electric field processing or enzymatic degradation, but this is always very cost intensive and industrial scale is questionable.
Yeah, thanks. Hopefully the participants are at least taking notes on this. But yeah, we can move to the next question. What are the different cultivation methods? What different cultivation methods can be used to obtain yellow colored biomass? Can we tweak cultivation parameters to produce different colors in microalgae?
So depends on the species that you use. Mainly heterotrophic cultivation, so under exclusion of light, using organic substances is used. Because you don't produce chlorophyll, they don't need it because they don't need photosynthesis. But there's also extensive research done on investigating microalgae that can be cultivated with limited amount of chlorophyll under photosynthetic conditions. Yeah. Or mixotrophic.
Okay. How do we deal with anti-nutrients and allergens while employing algae-derived components for human consumption?
Yeah, a very interesting question that I get quite often with the anti-nutrients or allergens. So to my or other people's knowledge that I've been talking about it, if you go for the approved microalgae, they haven't discovered allergens yet or anti-nutrients.
Interesting.
And we also wanted to conduct toxicology studies on it, but it's really hard doing studies there. I mean, there's cyanobacteria, probably the person who asked the question went for or thought of cyanobacteria, so algae blooms that can produce very toxic components, but we don't have them in those grass, so generally recognized as safe status microalgae.
Yeah, thanks for that, Iris. So Praveen is asking, is there a chance that force field electricity possibly could change the charge of amino acid residues in the active site domain? Perhaps there is a chance that the change in charge causes the protein to agglomerate with other proteins to precipitate down.
So using postelectric field processing as protein modification technology, right? Mm-hmm. Yes, we are also investigating that, how proteins behave in an electric field, how they accumulate or precipitate, or yeah, just form structures.
Thank you. Yeah, the next question is, instead of microalgae, are there any algae species or algal species that have been genetically modified to produce a target molecule, protein, so lipids, and then extracting, isolating the desired target for biomass production?
Yes, of course. So GMOs are also done for microalgae, but I think it's mainly for pharmaceutical, nutraceutical applications probably. But if we want to go for food production, we aim at not using GMO microalgae. But I've already seen research with people doing GMO microalgae.
Thanks, Iris. By the way, just to remind the participants that if your answer is not, I mean, if you think that your question is not answered completely, or you want to add something more to your question in terms of the question, please raise your hand and we'll unmute you towards the end of this session. 3 more questions to go. So the next question is, if extraction of phycocyanin is difficult in microalgae, I think there are 3 questions on phycocyanin. So if the extraction of phycocyanin is difficult in microalgae, can we engineer Saccharomyces cerevisiae with gene encoding alpha and beta subunits of phycocyanin? Yeah, the next 2 questions is probably about spirulina, so we can touch that later. But if you could answer to this question, if possible.
So extraction of phycocyanin, I mean, it's an intensive process too. So you need to use chemicals or do it at the isoelectric point and then separate. So it also depends on what you define as difficult, I think. But it's not just 5-minute reaction, because we also need to use ultrafiltration and diafiltration with the process.
Mm-hmm. Thanks for that. The next 2 questions are very similar. So what remains after extraction of phycocyanin from spirulina? Can the waste or the extract, I mean, the remaining part after removing phycocyanin, can it be used for something?
Yeah, and I think it should be, like I said before, we should use a cascade extraction, making use of all valuable components from the microalgae, which is crucial if we want to be sustainable. And eco-efficient, so to say. It was just for us, it was a study to see, okay, can we extract the phycocyanin and how does it compare to using the whole microalgae? Because for us, with the added nutritional value, it would always be a benefit using the whole microalgae biomass. But of course, one should also make use of the other components, lipids, vitamins, vitamin B12, for instance, from the spirulina.
Okay. Yeah, I think these were some of the questions. One last opportunity to raise your hands, participants. If you have any questions, shoot it out to Dr. Iris.
I just want to say, I was also late a bit, so we are delayed on the Q&A part. If you have questions, you can also contact me and I will try to answer the questions for the sake of being late for the meeting. So
absolutely. I mean, please don't apologize. I think we are living in time of a pandemic wherein this is quite common. So please, I think this is like, it happens all the time. So no worries about that. But yeah, please. Just
be free to also send me your questions then.
Yes, there is one person who is raising his hand. Ajay, if you can ask a question.
Hello, sir. Hello, ma'am. That was fantastic presentation from your side, and I got to learn a lot. Ma'am, my question is that you told about the cascade processing for the remaining part of the spirulina process. Using different chemical process during the extraction of phycocyanin from the spirulina, how it can be maintained the pure form while we are performing the cascade process for the remaining extraction?
So when I get the question right, is we extracted the phycocyanin, right? And the question is, how can we make use of the other components if you use the phycocyanin in this intensive process?
Yes, I mean, because we are already using different types of chemicals in the phycocyanin, extraction of phycocyanin.
Yeah, so, yeah, it's a very valid point. Probably, because we go, as I showed on the slide, from the crude extract to like the protein or soluble extract to the protein isolate, and probably we might need a processing step in between from going from the crude extract to the soluble extract, because there we are just soaking the microalgae or solubilizing them while we applied some pre-processing to it. And during this soaking period, so before applying the chemicals, we might already need to extract the other components. We are working, for instance, with microfluidics devices to test on the small scale, or filtration approaches, different solvents on how we can separate different components. But yeah, it's a fair point. If we apply very harsh chemicals, we might not be able to use other components.
Thank you very much for your answer.
Yeah, you're welcome. Thanks for the question. I think you are muted.
Sorry, yeah. I was just saying that thank you very much, Dr. Iris, as well as the participants, to make this webinar successful. And like Dr. Iris suggested, if the participants or any of you have any questions now, please shoot it out to us and we'll forward it to Dr. Iris, and you can talk to Dr. Iris directly after that. And I should take the liberty also to say that Dr. Iris will also be open to mentoring participants in phase 3 or teams in phase 3 who have specific scientific questions or technical bottlenecks or challenges in your idea, proposal, or your basic venture which you're starting together. But yeah, thank you once again, Dr. Iris, for making time out of your busy schedule, and this was an excellent presentation. By the way, for participants who are already here, Dr. Iris comes from a very, very reputed lab, Alexander Mathis lab. He's a very reputed researcher in food technology and food preservation. He has won bunch of awards. It's a long list, I cannot tell it here, but he's super aligned towards cultivated meat and alternative proteins. So I think everyone should leverage what expertise Dr. Iris has in this field. But let's end the webinar here, and I'll just stop the recording now. But thank you, everyone, and have a good evening.
Thank you.